Hello guys, and in the previous lecture we have discussed about the development of cortico-papillary osmotic gradient, and the two factors that were responsible for the generation of cortico-papillary osmotic gradient were: first is the loop of Henle as the countercurrent multiplier, okay, the loop of Henle as the countercurrent multiplier, and we have discussed about the descending limb and the ascending limb, how they contribute to the development of the medullary hyperosmolarity. And the second is the urea recycling mechanism in the inner medullary collecting tubules, how this absorbed or reabsorbed urea further contributes to the hyperosmolarity of the medullary interstitial fluid. We have discussed that. So let's revise a little bit, and then we will move towards the countercurrent exchanger, that is nothing but the vasa recta. So first of all, what is cortico-papillary osmotic gradient? As we know that when the blood moves from cortex to the medulla, it is less and slow. Okay, and nature has put a specialized type of capillaries in the medulla that are in which the blood is less in amount and slow in speed. What is the advantage of less and slow blood? It means that it will not wash away the solutes, okay, and the solutes are not washed away, it will contribute to the maintenance of the corticopapillary osmotic gradient. And as we know that when we move from cortex to the medulla in a particular nephron or in the kidney, we find hyperosmolarity in the medullary interstitial fluid. That is because the solutes are further concentrated in that medulla, and it is very necessary for the concentration or dilution properties of the urine. So let's talk about the maintaining part. The production part was, the first is the loop of Henle as countercurrent exchanger, and the second is the urea recycling mechanism, right? And how to maintain that particular corticopapillary osmotic gradient? This is by the specialized capillaries known as the vasa recta. Vasa recta arises from the efferent arterioles, okay, like the peritubular capillaries, but they are deep into the medulla and they maintain the medullary hyperosmolarity. Let's discuss about in detail. First of all, when blood move from cortex to medulla, okay, we are moving from cortex towards the medulla. This blood vessel is surrounded by progressively increasing hyperosmolar interstitium. Okay, as we move from cortex to medulla, the osmolarity is still increasing, right? Osmolarity is increasing, but the blood is passing progressively deep into the hyperosmolar interstitium, and the capillary walls are permeable to water and solutes. It means that when at this point, when there is surrounding hyperosmolarity, the blood starts to lose the water and gain the solutes. Okay, it will gain the solute, and when we move from cortex to medulla in the vasa recta, we will see that the vasa recta and the interstitial fluid starts to equilibrize with each other. It means that they start to equilibrate with each other, and when there is 300 milliosmol per liter inside the capillary, there is also 300. When 600, due to the gain of solutes and losing of water inside the vasa recta, there are also 600 milliosmol per liter. And we starts to see the osmolarity here at the converting point of the loop of the vasa recta. It means that when the medullary interstitium is around about 1200, inside of the vasa recta the osmolarity is same 1200, because there is gaining of solutes and losing of water because the capillary walls are permeable. When the counter movement starts, when the blood start to move from medulla towards the cortex, it starts to be surrounding by progressively decreasing hypoosmolar conditions. It means that uh, in in increasing hypoosmolarity, right? And as we know that capillary walls are permeable to water and solutes, so they starts to lose the solute and starts to gain the water, and they will become more hypoosmolar. And when the blood exit its osmolarity is around about 325. It means that it has only gained 25 amount of solutes. It has gained a very little portion of solutes. It is not responsible for the washing away the medullary interstitium. It has gained a very less amount of solutes, and that's exactly what we needed in case of the maintaining of the corticopapillary osmotic gradient. Let's discuss like revision type. When the blood is moving from cortex to the medulla, when the blood is moving in this direction, okay, the blood vessel starts to gain the solutes and lose the water and increase the hyperosmolarity. When it starts to move from cortex towards the medulla, it starts to gain the water and lose the solutes and become hypoosmolar, but not very much. It's like 325. It has gained only a bit of solutes. It has not washed away the medullary interstitial hyperosmolarity, and that's exactly what we needed in case of the maintaining of corticopapillary osmotic gradient. So what's the difference between countercurrent exchanger and countercurrent multiplier? The countercurrent multiplier refers to the loop of Henle, the descending part and the ascending part. The descending part is permeable to water and this starts to lose the water, and the tubular fluid starts to become hyperosmolar. It starts to equilibrate with the interstitial fluid. And in the ascending limb, lots of solutes are reabsorbed into the interstitium. They both multiply the capability of the system to generate the medullary hyperosmolarity. That is why we call them as the countercurrent multiplier. Countercurrent exchanger are vasa recta capillaries because these are just exchanging the water and solutes, and because their walls are permeable to both water and solutes and they gain a little amount of solutes at the exit point or around about 325, and they do not wash away the medullary interstitial hyperosmolarity. In case of countercurrent multiplier, there is active transport of solutes. As you know that in ascending part of the loop of Henle, there are special transporters which requires ATP for the reabsorption of solutes. But in case of vasa recta, there is passive transport of solutes. There is no requirement of ATP. Just capillary walls are permeable, so solutes and water starts to be transported passive, passively. In case of their function, countercurrent multipliers has responsibility for generating the osmolarity, medullary hyperosmolarity, but countercurrent exchanger are responsible for mainly maintaining the medullary hyperosmolarity, and we have discussed it in detail. Let's talk about the antidiuretic hormone. We have already discussed about ADH in the previous lectures. ADH is also known as the vasopressin. When we move from the ascending limb, the osmolarity is 1200. But a lot of solutes have been reabsorbed in this part, so osmolarity decreased to around about 100, right? And in the distal part, more specifically the early distal tubule, furthermore sodium and chloride are reabsorbed and the tubular fluid becomes even more hypoosmolar, and this part of the nephron, the distal tubule, is also known as the cortical diluting segment. Let's talk about the function of ADH. This is a principal cell. Okay, this is the principal cell of the late distal tubule. It can be present in the cortical collecting tubule and it can also be present in the medullary collecting tubule. ADH has special receptors known as the V2 receptors to be more specific, and these are the 7-pass receptors or serpentine receptors. These are joined with the G stimulatory. G stimulatory will stimulate adenylyl cyclase. It further converts ATP to cyclic AMP, and this cyclic AMP is responsible for the production or upregulation of protein kinase A. Protein kinase A is a phosphorylating enzyme. It phosphorylates certain transporters and it will cause the vesicle-loaded aquaporins to move towards the luminal membrane. In the absence of ADH, we see that the luminal membrane was not permeable to water. Okay, but in the presence of ADH, certainly it became permeable to water due to the insertion of the aquaporins into the luminal membrane with the help of protein kinase A. Although other type of aquaporins like 3 and 4 are present on the basolateral membrane, but they are of no function as soon as the water reabsorption is concerned from the lumen, right? So principal tubules and the late distal tubules and the collectican tubules, these all have principal cells in them, and ADH and aldosterone are functioning on these kind of principal cells. The function of ADH is to primarily retain the water, and function of aldosterone is to retain water and salt, mostly sodium. Okay, and in the presence of ADH, we know that a lot of water has been reabsorbed, so person will pass small amount of concentrated urine. And in the absence of ADH, when water has not been reabsorbed, it will cause the production of diluted urine in large amount. Okay, and for now, we have noted that ADH has function in the reabsorption of the water, but ADH has two important other functions. It stimulates the sodium potassium 2 chloride co-transporter in the ascending part of the loop of Henle, this pump. This pump is also ADH sensitive and it upregulates its activity, and it will cause more reabsorption of solutes, contributing to the medullary hyperosmolarity. And the second sensitive cells are the inner medullary collecting tubule cells. These are like special for the urea transporters, and urea transporters are sensitive to ADH. More urea is reabsorbed, it will contribute to the medullary hyperosmolarity. Okay, there is absence of water. So there are special type of receptors in the hypothalamus known as the osmoreceptors. These are osmolarity sensitive, and ADH is synthesized in the neurons of the supraoptic nucleus. In the neurons of the supraoptic nucleus, ADH is synthesized and it's packed inside the vesicles, and it is transported to the posterior pituitary by supraoptic hypophyseal tract. Remember that ADH is not directly produced from the posterior pituitary either. It is packed inside the vesicles. First, it is produced in the hypothalamus, then it is transported to the posterior pituitary to be secreted. When there is potential and release ADH. ADH's function in conserving the water reabsorption, and in this condition, person will pass small amount of concentrated urine. Okay, and when there is water, the osmoreceptors are not stimulated, and we do not need ADH. There will be inhibitory effect of ADH. Okay, there will be no ADH secretion, and person will automatically pass large amount of diluted urine. Okay, let's talk about the tubular fluid and plasma osmolarity ratio. What does it mean? It means that tubular fluid, the fluid present inside the lumen of the nephron, and the plasma, blood, their osmolarity ratio. What we get from the tubular fluid by plasma osmolarity ratio? First of all, in the Bowman space and in the proximal convoluted tubule, the osmolarity of the blood is 300, right? The blood has been filtered from the glomerular capillaries, and the tubular fluid in the PCT, right? In the proximal convoluted tubule, the reabsorption of water and solutes is iso-osmolar. It means that same quantity of solutes and same quantity of water has been reabsorbed in the PCT. So tubular fluid osmolarity is 290 still. it is 300 still, because the reabsorption is isoosmolar. So what will be the ratio of tubular fluid by plasma osmolarity? It will be 1. 300 by 300 is 1. Let's talk about the descending part of the loop of Henle, the descending limb. As we know that this part is permeable to water. When water is lost from the tubular fluid, the osmolarity starts to increase. Okay, osmolarity starts to increase, and but the osmolarity of the blood is still the same. So the ratio will be greater than 1. Okay, the ratio will be greater than 1 because tubular fluid osmolarity is increased. Talking about the thick ascending limb of loop of Henle, the lumen is permeable to solutes, but it is impermeable to water. It means that solutes will be reabsorbed, but water will stay in the lumen. It will cause the decrease in the osmolarity. Tubular fluid osmolarity will decrease and the plasma osmolarity will increase, and the ratio will be less than 1. In the distal part of the nephron, in the cortical collecting tubule, in the medullary collecting tubule, the osmolarity depends on the presence or absence of ADH. It means that when there is no ADH, in the absence of ADH, the tubular fluid osmolarity will even more decrease. Okay, in the absence of ADH, the tubular fluid is still hypoosmolar. Okay, it was 60, and in the absence of ADH there will be no water reabsorption. But in the presence of ADH, the tubular fluid osmolarity starts to increase because the tubular fluid starts to become more concentrated. Water has been reabsorbed, so it will start to become concentrated. Ratio will become 1 and then will become greater than 1 because the tubular fluid is more concentrated than the blood. So this was for today. I hope you understand the concept regarding the countercurrent exchangers and other concepts relating to the urine concentration and dilution properties. And I will see you in the next video. Till then, goodbye.